Good afternoon, everyone. Today, I would be presenting my research, which was to use machine learning approaches using the TCGA breast cancer data to fill in incomplete attribute characterizations. Datasets such as TCGA comprises of several instances, but however, they are not fully characterized. For example, breast cancer data set, which I've used for the study, for some samples, the estrogen, progesterone, and the HER2 receptor status column were labeled as not available, indeterminate, not evaluated, or equivocal. These receptor status is important in classifying breast cancer into different subtypes, as well as for its treatment. We aim to use machine learning as an approach to predict the receptor expression status of uncharacterized samples using a robust classification system. We used TCGA 2016 protein expression breast cancer dataset, and with the help of R, we obtained expression table as well as the phenotype table. The data was then divided into training set, which comprised of samples which had the receptor status column labeled as positive and negative, and the test set, which comprised of samples containing the receptor status column labeled as not available, equivocal, not evaluated, and undeterminate. So firstly, we used a training set to evaluate the performance of 6 different classifiers, which were J48, random forest, random tree, SMO, naive space, and multi-layer perceptron. Now, since the number of receptor positive and receptor negative samples were unequal, to balance the sample size, we used median supplement method. And after evaluating the performance using a 10-fold cross-validation technique, we chose J48 due to its high precision and sensitivity across all the 3 receptors. So we used J48 to predict the test set, and after predicting the test set, we classified training and test set into non-triple negative breast cancer and triple negative breast cancer groups. To validate the predicted results, we performed 4 different methods. Firstly, we used a bioconductor package called SIG to identify proteins that were differentially expressed between the non-triple negative breast cancer and the triple negative breast cancer groups in training as well as test set. We found 65 differentially expressed proteins and 19 differentially expressed proteins in training and test set respectively, out of which 8 differentially expressed proteins were common to both the sets. Figure 2 represents heat map of differentially expressed proteins in training set and the test set. The red represents that a protein is upregulated in the triple negative breast cancer group, while the blue represents that the protein is downregulated in the triple negative breast cancer group. Then we use these differentially expressed proteins, and using another bioconductor package called Reactive PA, we determined the overrepresented pathways. We found that activation of MAPK pathway to be common in both of them. Additionally, this was confirmed when we mapped the differentially expressed proteins on cytoscape to observe protein-protein interactions. The figure 3 represents the protein-protein interactions between MAPK family and HER2 protein in training as well as test set. Hence, this draws the result that the test set is similar to the training set. Furthermore, we also used a tool in Cytoscape called J-active modules that predicts the protein-protein interaction based on the p-value. We found physical interactions between these proteins in both the sets, and on further research, these could be potentially target for treatment of triple negative breast cancer. Here as well, the red represents that the protein is upregulated in triple negative breast cancer, while the yellow represents that the protein is downregulated in triple negative breast cancer group. Green represents that the protein is not differentially expressed between the two groups. Lastly, we performed principal component analysis to determine the correlation as well as directionality of these proteins. We observed VEGF-R2 to be highly correlated with ER alpha in both the sets. Also, it was confirmed the correlation between FOXM1, EGFR, and CDK1, which was observed in the test set using cytoscape, to be similar in the test set using principal component analysis. In conclusion, we were able to characterize and validate the receptor status in TCGA breast cancer dataset using machine learning approaches. We also were able to find protein-protein interactions and those proteins which were upregulated in triple negative breast cancer that could be potentially targeted for the treatment of breast cancer. Thank you.